Welcome back to Principles of Organismal Biology. Before we jump into the class, I just want to remind everybody that the midterm is coming and that you need to prepare for it very good. So good luck with it. The midterm is going to be at noon this Friday, September 18th, and it's going to be synchronous. It's going to start sharp at noon, so make sure to log in into your Canvas to have access to the exam. And it's going to cover everything from the very first class until the last class, which is today's class. And that includes prokaryotes. You need to complete modules 8 and 9 and also complete the pre-test exam before the midterm. Good luck, and please read and prepare yourself carefully for this exam. Last class, we learned about phylogenetics, how to read phylogenetic trees, the tools that we use to reconstruct evolutionary relationships in the history of the species. Today's class, we're going to jump now into understand diversity in different groups. And the group that we're going to review today is prokaryotes. This one here is not a prokaryote, but rather is a Hawaiian bobtail squid associated with some very cool bacteria. These bacteria are bioluminescent, and they can glow at night when this squid is active and is trying to hunt something. But then what happens is that in absence of these bacteria, the squid will cast a shadow that then now predators can see and hunt the squid. Now, by having this association with this bacteria, the bacteria will glow, and then the predators will no longer see the squid, right? Because the bacteria are somehow mimicking the light coming from the moon. I think it's a very cool association between animals and bacteria, and they are also very gorgeous looking. These are the goals for today's class. We're gonna review horizontal gene transfer, identify the two main groups that make prokaryotes, that's archaea and bacteria, and we're gonna understand and learn some structures in this group that are important for motility, reproduction, and nutrition. And we're gonna review or present some of the clades and functions of bacteria and archaea as well. We presented last class that horizontal gene transfer is the movement of genes from one species to another one, or from one genome to another. And this is facilitated sometimes by transposable elements in plasmids. We're gonna learn today about plasmids, viral infections, of course, and sometimes the fusion of organisms, such as what happened with the endosymbiosis of mitochondria and the chloroplast, right? We have reviewed that last class. So this creates a very messy pattern in the evolutionary history of this species, especially early on in the origin of life. That is hard for us to reconstruct the phylogeny. And we say that rather this phylogeny looks more like a network, like in here. Horizontal gene transfer can be very prevalent in bacteria. In this example, we have one bacteria, which is the donor, and this at the bottom we have a receptor bacteria. And you see how we are having transferring DNA material from one bacteria into the other one. So that's also horizontal gene transfer, and we're gonna learn a little bit more about it today. Now, do you think that humans have genes in their genomes product of lateral gene transfer? Yes or no? This is a poll. Okay, well, the answer is yes, and I'm presenting here two different examples. One is in the human papillomavirus, HPV, which causes cervical cancer. And we have here how it looks in the cervix. These cancerous cells are growing. They now have altered the cell cycle, and they just go crazy with mitosis and duplicating their material. So this is associated to the presence of this viral DNA. So virus can transfer DNA, pieces of DNA into human cells that result in these cancerous cells. Another example happens with the Helicobacter pylori bacteria and the Epstein-Barr virus. Both of them can cause gastric cancer, or at least have been associated to gastric cancer. And in here, we're representing the bacteria, and the DNA is circular, it's blue, and a chromosome in humans, right? These X type of chromosomes that we have. And then a chromosome acquires, human chromosome can acquire a piece of the DNA from this bacteria, from Helicobacter pylori. And when we sequence this piece of DNA, we can see that this chunk of sequence is coming from the viral DNA. So yes, humans also experience horizontal gene transfer. Now, I want to ask you something. Have you ever feel lonely? I think so, right? Everybody has felt lonely occasionally. What I want to tell you is that you shouldn't feel lonely, because we are actually a community, and you have a lot of thousands of bacteria in your body, and at least 500 to 1,000 species of bacteria associated in your guts. So that's actually very, very interesting, and maybe new for some of you. And that's not even, it doesn't stop there. If we look at the proportion of cells, not the size of the cells, but the number of cells, it turns out that human cells are only a third, and the rest is bacterial cells, or viral, or fungal. And if we talk about bacteria, we refer to the microbiome, the virus make the virome, and the fungal will make the mycobiome. So you see, we have a lot of things going on with us. Every time we move, we carry all of these species with us. And also we have little insects, right? Mites that come out of our pores at night and eat our lipids in our face, or other type of little tiny insects that we carry around. Yes, we do. Yes, we do. Bacteria can be beneficial and harmful. So it's very important to recognize we have different types of bacteria, and actually a lot of them are beneficial. And it will depend on what our diet is and our practices. If we exercise, the type of bacteria that we can develop in our body. For example, in here we can locate different types of bacteria that associate to different parts of our body. And that is part of this study of the microbiome, the human microbiome. And if we look at the guts, there are a series of bacteria, and some of them, depending on the substrate that they are going to be breaking down, they can be beneficial or harmful. And in here we have this tryptophan, which is an amino acid, and in the presence of different bacteria, this tryptophan is going to be converted into a different molecule. For example, Clostridium sporogens, it's going to convert this tryptophan into IPA, which is the indolopropionic acid. And this acid gets inside, so it passes the epithelium in the guts, and then it gets transferred into the brain. And in the brain, it can have some beneficial activities, such as it reduces DNA damage. It can act as an antioxidant. And more importantly, I think, or very excitingly, it inhibits the beta amyloid fibril formation, which is actually associated to Alzheimer. So with the inhibition of this will reduce Alzheimer. You see, and this is just the presence of this bacteria, which is leading to all of these positive effects. These other bacteria, in contrast, tryptophanase expressing bacteria, can convert this tryptophan into indole, and this indole can have some damage to the liver and kidneys. So it really depends on the bacteria, the molecules that we have in our organism, the type of responses. And isn't it fascinating that some of these bacteria can even act or have an effect in the neurotransmitters that go into our brain. And it has been associated, the presence of some bacteria now have been associated to our mood. And that's very important and interesting to recognize that. In here, I have a picture of Dr. Erika Forsberg. She studies these type of associations with the microbiome and the human gut. So if you are interested in some of her research, please contact her and you might be able to join her lab, who knows. In here, I have a picture of Dr. Scott Kelly. He is a professor and a very nice colleague in our department in biology who studies the human microbiome. In here we have, this representation. This is a phylogenetic tree as well that is now represented in this type of spiral shape. But still we identify some groups in here and other groups in here that are more closely related. Let's take a look to this. This is Staphylococcus aureus. This is the one that colonizes our skin, and it's not harmful as long as we don't have cuts or scratch in our body, or we don't go through a surgery and we get exposed to this Staphylococcus. Remember, this Staphylococcus aureus was very important, was an example that we use to understand natural selection. And some of these bacteria have developed antibiotic resistance. And that's the reason why we look into that example. Hopefully you remember that. And then you can see in this phylogeny, the different type of species and their location in our body. For example, this Campylobacter has pathogens that can also be present in our mouth. But there are other more beneficial bacteria that we can also find in our body. So we are not the only ones that interact with bacteria. In this experiment, you can see plants interacting with bacteria, and these bacteria are beneficial for them. This experiment consists of growing plants with soil that has bacteria and soil that doesn't have bacteria, like here. So the uptake of potassium by plants is measured in the y-axis. And you can see that when there is no bacteria in the soil, this type of beneficial bacteria, not any type, the uptake of potassium is lower versus when we have this strain of bacteria. So some of the bacteria actually can be more beneficial to the uptake of nutrients and minerals in the soil. Now think about this. Prokaryotes are everywhere, really everywhere. You can find them in very extreme places, in places that are very acidic or salty, cold, or very, very hot. And these might not be suitable for all of the species, but prokaryotes can be thrive in these places. And because this ability, they are very abundant on Earth. Prokaryotes divide in two domains, bacteria and archaea. We keep saying that, and I hope that by now you remember that. Prokaryotes were the first organisms inhabiting Earth. They are unicellular, and some of them can form colonies. They are smaller than eukaryotes. We can see here in this figure the representation of the atom, etc. Then we have virus, and bacteria, along with mitochondria, have this length or this size, which is 0.5 to 5 micrometers, which is 0.5 to 5 micrometers. And then we have the eukaryotic cells over here, the egg human, etc., until adult female. So you can have this comparison of the sizes. And it's important to remember prokaryotes are smaller compared to eukaryotes. Most common shapes of prokaryotes are divided in these three forms, which are spherical, also called coccus or cocci in plural, These rod shape are bacillus or bacilli in plural, and the spiral, which is just spirals in plural. It's very easy, that one. Now, we say that the prokaryotes are going to be divided in 2 domains. So we have, in this tree of life, we have 3 domains. The domain eukarya, we're going to cover later. And today we're covering these 2 domains, domain archaea and domain bacteria. Those belong to this artificial group prokaryotes, right? Because they are not a natural group or monophyletic group, but rather they are a paraphyletic group, referring that this group has the ancestor and some descendants, but not all, because the ancestor of the most common recent ancestor of bacteria and archaea is right here. And from this ancestor, we also have eukarya, but it's not included in our definition of prokaryotes, right? Now, archaea, there are several features that distinguish archaea and bacteria. And this phylogeny has been constructed using ribosomal RNA. So genomic information separates bacteria and archaea. But there are other features that also separate these groups. Archaea is gonna lack peptidoglycans in their cell wall. And bacteria are gonna have peptidoglycans. And this is a network of sugars, these polymers interlinked or cross-linked, and create a structure that help this bacteria preventing burst. Now, in bacteria, we are gonna have gram-positive and gram-negative bacteria. Maybe you have heard about this. Gram-positive are gonna be bacteria that have this peptidoglycan layer in the outside. So this is close-up of the bacteria, the plasma membrane only. And in the outside, we have this thick peptidoglycan layer. That's the cell wall. In a gram-negative bacteria, we're still talking about bacteria, we will have this type of sandwich approach in which you have peptidoglycans. This is important to remember. This one down here has peptidoglycans in between these 2 layers of plasma membrane and the outer membrane. So the cell wall then in this case is combined into these 2 elements, and the peptidoglycan and the outer membrane in the gram-negative bacteria. So gram-negative bacteria have peptidoglycans, but they are not in the outer layer. So when we use some chemicals to stain these peptidoglycans, the gram-positive bacteria are gonna turn purple right here. Whereas the gram-negative, the sandwich ones, are gonna turn pink. Now this is a very important structure, the cell wall, because the antibiotics that we produce usually targets the formation of the cell wall. So the cell wall in bacteria is replenishing constantly. So it's renewing the cell wall. And then when you inhibit the formation of the cell wall, what is gonna happen is that now the bacteria is susceptible to homeostasis, right? And then the bacteria can burst if you have more solutes in the inside than outside. Another structure that is important is this capsule. And the capsule can be formed from polysaccharides and also proteins. And in this figure, you can see the capsule. And the capsule will be formed outside of the cell wall. And this capsule can be used by the bacteria to adhere to the substrate, and to the substrate or to other individuals of bacteria, with other bacterium. And these can also provide a shield to pathogenic bacteria from the host immune system. This is a very important structure for the bacteria, this capsule. In our body, we can have, the tonsil cells, right here, the tonsils, and these bacteria can adhere to our cells using this capsule. Another structure is the endospore. The endospore is formed sometimes by bacteria when they need to become dormant. And that means that they're going to reduce the metabolic rate in this species, and they form this endospore. So in here you can see the endospore, and they can remain dormant for centuries, even centuries. The other thing that is important to consider is that this endospore will allow them to survive higher temperature, lower temperature, or other harsh conditions. This is the structures that are considered appendages, and we're going to have 3 different types of appendages. The fimbriae here are type of hair-like structures that help bacteria to anchor or to attach to some structures because they are sticky, and they can attach to structures or to attach to other bacterium when they are forming colonies. Pili or pilus in singular, I know it's counterintuitive, but pilus is singular or of pili or pilii. This structure, the pilus, is gonna be providing a mechanism to exchange DNA, and we're gonna cover it a little bit more, but this is very important to exchange DNA between different bacteria. So right here is the pilus, and you can imagine DNA going around this pilus and getting transferred into these bacteria over here. Lastly, we have the flagella or flagellum. Flagellum singular and flagella is plural. And this is a very common structure that facilitates movement in these bacteria. What I find fascinating is that this structure completely deassembles. Like, look at my hand, I can turn it, but it doesn't completely move around. Just think in the, what is that movie called? The Exorcist, with the girl that turns around the head. We cannot do that. It's very uncommon in nature that we see that, but bacteria can actually completely turn around this structure, the flagellum. And they can reach very, very fast speed. And these bacteria or prokaryotes, because also archaea have this ability to have flagellum, they can have phototaxis or some sort of taxis, which is that they can move to towards or away from a stimulus. That's what taxis mean. And chemiotaxis, for example, means that they move towards or away from a chemical. Let's take a look to this short video for you to see how fast bacteria can be. And you might not be impressed because these are tiny little creatures, and you can see the flagellum right here. Wow. But then these bacteria or these prokaryotes can have a speed of 50 micrometers per second or higher. And this is the comparison in size will be comparable to a Ferrari, these speed cars that can be super, super fast, like, I don't know, maybe 300 kilometers per hour. And in these guys can beat these Ferraris, okay? So that's for you to make the comparison. And now, prokaryotic cells lack nucleus, right? We say prokaryotes means pro, before, and karyo, that comes from karyon, that's Greek, and that means before the nucleus or before the nut, before the nucleus. And they lack this compartmentalization, not only related to the membrane of the nucleus or the nuclear envelope, but also regarding to all of the other compartments that we see in the eukaryotic cells. So they lack compartmentalization. However, some bacteria have these specialized membranes that they have in folds, and in some aerobic prokaryotes and of bacteria, we have this respiratory membrane. And in others, we have this type of thylakoid membranes. These ones are associated with photosynthesis, and these with aerobic respiration. These remind us of the mitochondria, right? And this one, the thylakoid membranes, that are able to photosynthesize remind us of the chloroplast in plants or in algae. Prokaryotic DNA, if you remember our class on genome evolution, you probably remember that prokaryotes have from 1 to 6 million base pairs. And then we have eukaryotes that can reach to 35 gigabase pairs, which is a lot more. Other difference is not only the size with mothers, but the actual structure of the DNA. In eukaryotes, we have linear chromosomes, whereas in prokaryotes, we have a circular chromosome, one circular chromosome. And the chromosome is not going to be contained in this nucleus. As I mentioned before, prokaryotes like the nucleus, but they have a nucleoid and plasmids, several plasmids, not only one. So here we have the circular chromosome, which is having some loops, so you cannot see the circle, but it's just a circle that it's folded. And this area in here, it's called the nucleoid, containing this containing this chromosome. And then we're gonna also have small pieces of DNA called plasmids. And these are right here, okay? So what is this called? Flagella. And what about these little hairs in here? Fimbriae, okay, good. Now, what is also important to mention is that this chromosome in prokaryotes lacks histones. And that's something that we learned early on in our second class, that chromosomes in eukaryotes have histones and also help with this condensation of the chromatin. The reproduction of prokaryotes is really fast, and they can have division of the cells in 20 minutes, even less, but usually they divide in 1 to 3 hours in optimal conditions. And these are very key features for their biology. They are small, they have binary fission, and that's actually the way that they divide is not through mitotic process. And also, obviously, they don't have meiosis either. They have a simple version of that, which is called binary fission. And then in this case, both this large circular chromosome and the plasmids are gonna duplicate or replicate. And after that, they migrate to 1H, and then the cell, the membrane just synthesize, and then they just fuse in two different cells. They have shorter generation times, and they also have genetic diversity, high genetic diversity, and that is associated to this rapid reproduction that we just looked into, but also associated to mutation and genetic recombination. Regarding to mutations are actually low in these prokaryotes, but because the reproduction is really, really fast, like 20 minutes, let's say, and you have in 1 year a lot of generations of these bacteria, then yeah, the mutations can accumulate. And that's why we have a lot of diversity. And also because the population size is very large. So they reproduce asexually. They reproduce asexually through this binary fission. And they tend to be identical, but we'll see soon that sometimes we have other processes making this genetic diversity increasing. We're gonna have different sources of recombination in prokaryotes, and that is when we combine DNA from two different sources. And in this case, in prokaryotic cells, we have transformation, transduction, and conjugation. So let's look at these different forms. In this example here, we have a bacteria that bursts, and the DNA gets chopped into small pieces. And one of these small pieces can just get inside of this other bacteria, and it is incorporated later into the chromosome of this original bacteria right here. So that is called transformation. It's just very simple. It's the simplest way to acquire foreign DNA in prokaryotes, by just swallowing a piece of DNA inside of the cell, and then it gets ligated into this chromosome. The second mechanism is transduction. Let me move this over here. So in transduction, we have movements from different bacteria, but using phages. And hopefully you remember that phages are viruses that attack bacteria. So they're called also bacteriophages. Let's look into this example. Here we have a donor cell, and this donor cell is being affected by or infected by this phage. This phage is injecting the DNA, and just remember, the phages use these bacteria to turn them into a factory of phages. So now that the DNA of the phage is inside of this donor cell, this bacteria, the bacteria is going to make all of the pieces to assemble many bacteriophages now. So now that they get assembled, we also have in the bacteriophages, they are going to have copies of the DNA, and then the DNA get encapsulated in this virus, right? A piece of the DNA from the original bacteria, from the donor. And now when this bacteriophage is attacking another bacteria, it can donate this piece of DNA. And if you see, we have now this area that can recombine, or it can have crossing over, which with this circular chromosome of the recipient cell. And now it will look like this, a recombinant chromosome, right, with a piece of the original bacteria over here. Are bacteriophages a particular type of bacteria? Yes or no? Well, they are not, right? Sometimes my students get confused, but they are not. They are virus. They are very different. If you are interested in virus and bacteriophages in particular, Dr. Dwayne Roach and Dr. Forrest Rower work in this in the biology department, and I will suggest you to contact them as well. The last mechanism by which bacteria or prokaryotes can exchange DNA is conjugation. And this is when the pilus, the structure that we just learned, comes handy, because look at this. This is the pilus, and the DNA is going to be transferred using this pilus between this bacteria into this one. And in this case, we have this mating bridge, which it's made with the pilus. The plasmid, do you remember that I said that bacteria or prokaryotes have chromosomes, circular chromosomes, the main one, and also smaller pieces called plasmids? Well, these plasmids in here have two strands. And if one strand breaks, then one of these strands can migrate through this pilus into the recipient cell. And in there, once that it migrates, it forms its circular shape. And this new cell can help the plasmid to form the complementary strand of the strand that it moved into this recipient cell. So this is another mechanism by which prokaryotes can have a type of exchange of DNA. And that really explains why this group has a lot of horizontal gene transfer, especially early on during the origin of life. It's hard for us to reconstruct what happened right here because of all of this exchange of DNA happening, and that still happens. Now, please answer what is true about prokaryotes. A, lack a membrane-bound nucleus. B, lack mitochondria and any other membrane-bound organelle. C, the word prokaryote comes from the Greek pro before and karyon not or kernel, also nucleus. D, it is an artificial group, or all of the above. Okay, it's super easy, right? It's all of the above. All of these features are something that characterize prokaryotes. And it's very important for you to understand that this is an artificial group because it's not monophyletic. And if you forgot about the circular chromosome and the lack of envelope, nuclear envelope, well, this is here. Now, the origin of prokaryotes dates back to 3.5 billion years ago. And there are new studies that suggest that that's even older, like 3.8 billion years ago, which is a lot of time. And that has led to an incredible diversity in prokaryotes. And that includes both archaea and bacteria, this artificial group of prokaryotes. And if you just pinch soil, or you have a little bit of soil in your hand, you will have more than 1,000 prokaryotic species. And just imagine all of the possibilities to exchange genes among them. So that's crazy. And okay, let's look at the diversity in these groups. Bacteria, over here in our phylogenetic tree, is the group that has peptidoglycans, either as gram-positive, right here, or all of these gram-negative. Remember, archaea lacks peptidoglycans. It has a cell wall, but it's not made out of peptidoglycans. Let's review this. Do you remember how to read phylogenetic trees? Now, let's find the position in the tree for the last common ancestor of bacteria. Is it eukarya and archaea? Maybe TAC? Maybe 3 and 4, or number 3, ancestral, universal ancestor. What is the ancestor of bacteria? The last common ancestor of all of these bacteria is right here. Okay, hopefully that's clear for you. And if not, come to my office hours. Also gonna have different type of nutritional types in bacteria. Some of them can be photoautotrophs, others can be chemioautotrophs or heterotrophs. And just remember, the type of nutrition refers to where the carbon or the type of inorganic molecule, the molecule they are using to store the energy is coming from. If they made it, these are autotrophs. They can be photoautotrophs or chemioautotrophs, or just called autotrophs. But if they are sourcing their carbon from somewhere else, they are heterotrophs. And if they use light for this, for their metabolism, they are called photoheterotrophs. If they are using other type of chemical energy from inorganic oxidation, they are called chemioheterotrophs, or if not, just heterotrophs. So bacteria has these 3 forms: photoautotrophs, chemioautotrophs, and heterotrophs. This is the diversity in bacteria. We're gonna go through each of them, and we're gonna look into protobacteria, chlamydias, spirochetes, cyanobacteria, gram-positive bacteria. And this, if we zoom into protobacteria, inside of this lineage, we can have these relationships. We will have alpha protobacteria, beta, gamma, delta, and epsilon protobacteria. We're going to cover some of them in this class, not all of them, but alpha protobacteria have a couple of very important members. So in here we have rhizobium, and rhizobium, it's very interesting and super cool with plants. It's a cool body of plants because that bacteria can fix nitrogen, atmospheric nitrogen. And I think nitrogen is in the air, around 78% is nitrogen. But that form of nitrogen is not good for plants, or well, it's not usable for plants. So these bacteria can convert this nitrogen into another type of nitrogen that now can be used in the plant. And these bacteria live associated with the plants, in the roots of the plants, and in there is that they have these conversions of the nitrogen. So now the plant can use the good type of nitrogen and use it to build amino acids or build nucleic acids, which are high in nitrogen, right? Another example of alpha protobacteria is this agrobacterium, which has been heavily used in genetic engineering because its ability to produce tumors in plants. Now, another group of protobacteria, let's skip beta and jump into gamma. There are some sulfur bacteria, such as Thiomargarita namibiensis, which is the largest bacteria. I think it's 750 micrometers long. But there are others that we are very familiar with, such as the Salmonella and Vibrio cholera, which are pathogenic bacteria for us. And yeah, we can get in serious troubles if we catch some of them. One that is very common is Escherichia coli or coli, because that is associated with the intestines in humans, when also in many mammals, and is normally not pathogenic. So it's found in very small percentage and not very pathogenic, but when it's in the higher numbers, it can also cause some issues. These are examples of protobacteria that are obviously gram-negative. And Escherichia coli has been used heavily in science. A lot of people in the biology department use E. coli, and that was the one used by Martha Haish and Hershey in their blender experiment. Hopefully you remember that. Now, let's skip on delta and jump into epsilon. In this group of protobacteria, we have Campylobacter or Helicobacter pylori, the one that I mentioned that has been transferring DNA into cells in the cells in the stomach cells that can cause cancer as well. So this is associated with the stomach ulcers, Helicobacter pylori. Now, what's the sister clade of chlamydias? A, protobacteria, B, spirochetes, C, both protobacteria and spirochetes, D, cyanobacteria, or none of the above. Okay, let's take a look. So chlamydias are right here. Let's go back to the most common recent ancestor is right here. What is the sister? Oops. The sister is right here, spirochetes, okay? Chlamydias is this group of parasitic bacteria that are found in many animals. And we could have Chlamydia trachomatis causes blindness and non-gonococcal urethritis and by sexual transmission. And it can be very painful if you have chlamydia and you have to urinate or pee. You can have some issues there. Okay, so better to take care of yourself and not meet this guy. Another type of bacteria that has this very cool shape, these spirochetes, have this spiral form. And they are helical gram-negative, and they are heterotrophs, so they are not, they need to outsource their form of energetic carbon. Examples of that are Treponema pallidum, which causes syphilis, and also another bacteria, Borrelia burgdorferi, causes the Lyme disease. This is important to know that the vector are actually ticks, but the actual disease is caused by this spirochete. The next group of these bacteria, so we have covered all of these gram-negative, right, protobacteria, chlamydia, spirochetes. Now let's jump into the cyanobacteria, another gram-negative, which is photoautotroph. That means that they are going to fix their carbon and they're going to build their own molecules storing the energy, and they produce, as a byproduct, they produce oxygen. It's just like the chloroplast in plants, and remember this hypothesis in our theory of endosymbiosis or serial endosymbiosis, which explains that cyanobacteria are actually the ancestors of chloroplasts in plants. And cyanobacteria are very, very abundant in the fresh water and in marine plankton, and are contributing a lot to the oxygen that we are breathing right now. The last group of bacteria is these gram-positive bacteria. Remember, those are the ones that have the peptidoglycans in the outer layer. They sometimes can form colonies, and some of them are called actinomycetes, and they can help to decompose organic matter. Some of them, we have seen them and talked about them before, such as the Staphylococcus aureus or Streptococcus. And this is the flesh-eating bacteria that you might remember from our classes before. Other examples are Bacillus anthracis that causes the anthrax disease, or Clostridium botulinum, which causes botulism. Your book says that early evolving groups, they refer as basal groups, which are the group that evolved earlier. But we actually try to avoid talking about them as basal groups because there is nothing as basic versus more advanced in evolution. So what is the early evolving group of archaea? Prokaryotes. So that would be B. We have covered bacteria, and now we're going to learn about archaea. And a lot of them, but not all of them, like to live in extreme environments. And for example, we have archaea in our body, but the proportion is very small of the archaea that we have compared to bacteria. Most of the archaea like to live in extreme places, such as the halophytes, which live in extreme, highly saline environments, or the extremophiles, which thrive in very hot environments. And when I say hot, it's really hot. They can thrive in like 120 degrees Celsius, which I think is around 250 Fahrenheit degrees. So imagine, we just had a heat wave and I was so hot with, what was that, 90 degrees, almost 100 degrees in some places. That's super hot for us. These guys can handle this and more without any trouble. This is the same picture, but it's to elaborate more into the very salty environments that they can handle. This is archaea from the genus Halobacterium, which actually in very high conditions, in very dense proportions, they can turn the blue water into pink water because they are actually pink. And this is a picture of Laguna Salada de Torrevieja in Spain. And maybe we can see one day this. I haven't traveled there to see this by myself, but it looks quite interesting. Another group in archaea are the methanogens. And these methanogens like to live in swamps or marshes. They produce methane as a waste product on their metabolism. These methanogens are anaerobic. So that means that they will be poisoned if they are exposed to oxygen. And you can find them in the rumen, like in the stomach of the cows, right? These ruminants, these type of cows have methanogens, and they actually help with the digestion of the grasses that these cows eat. Now, they are going to be producing methane. And if you have watched Borat, I think Borat, there is a scene in which you can see a cow exploiting just because the formation of a lot of methane that accumulated in this cow. Anyways, not a fun scene to watch, but if you have watched it, you know what I'm talking about. And if not, you should watch the movie. Okay, this is a comparison of the different domains. We have here the domain bacteria, archaea, and eukarya. And these are different characteristics associated to the domains. So let's please start reading about this and carefully, and you'll find that there are sometimes the domain bacteria and archaea share these traits, like they lack nuclear envelope, and they also like these membrane enclosed organelles. And they also share having a circular chromosome, whereas in eukarya, we have talked that we have linear chromosomes. Now, if we compare the domain eukarya and archaea, we can find that there are a lot of commonalities as well. So they share having peptidoglycan, the absence of a peptidoglycan in the cell wall, whereas bacteria are the only ones that have the peptidoglycans. They also have several kinds of RNA polymerase. They are going to have the methionine as the initiator of the amino acid for the protein synthesis. And this is what I told you, AUG is the codon codifying for this methionine, right? Whereas in bacteria, it's gonna be as similar, but not identical amino acid. That's the formyl methionine. Another commonality or share feature between archaea and eukarya is that the growth is not inhibited when using these antibiotics, streptomycin and chloramphenicol. But it is inhibited in these bacteria, in these groups, in most of them. Okay, so, and then you can see the shared features between bacteria and eukarya. And again, you can imagine that this is hard for us to puzzle or put together what traits to use when we reconstruct these phylogenetic relationships in between the domains. And again, this is probably likely due to all the horizontal gene transfer that has occurred through the history of life that mask these linear trajectories, and that is because life is not in one direction. Yeah, in purple we can see the comparison between bacteria and eukarya, what are longer than fimbriae and allow prokaryotes to exchange DNA, A, B, C, D, or E. And the right answer is right there. That's Pili. Pili is the right answer. Okay, that's D. Okay, I hope that you like this class. This is a lot of fun. We keep learning about archaea. We don't have that much information about archaea because it's a very difficult group to study because they live in very extreme environments, and also we just have discovered a lot of these recently. But if you want to learn about methanogens, don't forget to contact Dr. Marina Kaloshnaya. She is an expert in that topic, and she'll be super happy to talk to you. Anyways, the exam is coming. Please read, study, and very good luck to all of you in the exam. I hope that you have learned a lot of things in this first part of the class, and I'm looking forward to see you in my office hours. Take care.